I had almost a page of information for introduction, and Sir Richard Roberts told me that I should just say so much of it, because he's going to talk more about his biography for you. So there are just two things I want to say. One was that as I was reminiscing about New England Biolabs, or NEB, we were chatting where he is now and has been for many years, how NEB was our Bible as we were in graduate school. The catalogs were better than any book available, and their enzymes were magical. So we are thankful to all his leadership in that company. And the second thing I just wanted to brush on was that his discovery of split genes, and he'll detail that more for you, was truly a paradigm shift. In fact, it was a tectonic paradigm shift. And he will go into it because the dogma at the time was that a unit of hereditary information that we call a gene the dogma was that it was continuous. So their findings were showing that it was not, that these genes were split. They were split genes. And they did that in adenovirus, but eventually we have seen that most life forms have split genes, including us, and that how those split gene information is edited is very important. And it's important for normal development and for, as well as for health. So for their seminal discovery of split genes, Sir Richard Roberts and Dr. Philip Sharp shared the 1993 Nobel Prize in Physiology or Medicine. So please help me in welcoming Sir Richard Roberts.
Well, thank you very much. It's a pleasure to be here. Nice to see that there are lots of you out there. We have a little bit of reverberation here. Is that something you can fix, perhaps?
If we turn that one off, does it work?
Well, we could see. Is that better now? Is it gone? Maybe you turned this off, did you? Perhaps that's good. I tend to walk when I talk, and there is no clock at the back, so I'm going to maybe walk around here a little bit. I have a clock up here, which if I don't see the one over here, so I will do this to keep in time. But it's always a mistake. If any of you ever get asked to set up a lecture theatre, always put the clock at the back so the speaker can see it, and never at the front where the students can see it, because they're just interested in when it's all going to end so they can get on with the next thing. So what I will try to do during the course of this talk is to give you some idea of how I came to win the Nobel Prize, kinds of things that I found to be important. And I'm hoping that along the way, I can highlight one or two of the things that I learned as a result of being a student and following this path that I think can really be useful to a lot of people and to a lot of you. And I have a little summary of this at the end, but I'll try to go into some of this along the way. Now, I grew up in England. I was born over here in the town of Derby. When I was 4 years old, my parents moved to the town of Bath in the southwest. Derby is not a particularly nice city, although it's a lot nicer now than it was. I was born during the Second World War, and the Rolls-Royce factories that were making all the engines for the Spitfires that the RAF were using to go and bomb Germany were being made in a factory about 10 miles away from where I grew up. And as a result, there were German bombing campaigns coming in all the time, and more than half the houses in the street in which I lived had bomb damage. There was a church over the way that had bomb damage, and so it was not the nicest of places. Bath, on the other hand, is lovely. If any of you ever go to England and you're looking for a nice place to go and visit after London, then I suggest you take the train to Bath. It's about 2 hours. You can spend a day there, walk around. It's just a beautiful, beautiful city. So I had a really great childhood in Bath, and I'll tell you a little bit about it. So one of the things that happened when I was quite young was I went to St. Stephen's School, shown up here. Very small school. It had 4 classes, about 25 kids in each class, 25, 30 kids in each class. And it was just an absolutely typical small-town school. It was not religious, even though it was called St. Stephen's, but of course, you know, in England, all of the schools ended up getting these Christian names associated with them. But I discovered a lot of things here, but in particular, the headmaster turned out to have a tremendous influence on my life, one that at the time I didn't really appreciate, but later did. And he must have seen something in me, I don't know what it was, but he used to come after school and would give me puzzles to do. And usually these were little mathematical puzzles, and I just loved them. I fell in love with puzzles and games, and I fell in love with mathematics. And just as a result of this one teacher who probably had a greater influence on me than anybody else who I ran into. By age 11, I read a book. My father had bought me this book, which said How I Became a Detective. And it was sort of a story of a detective, and it sounded fantastic. I mean, this was one of these things. Here you are, you're presented with crime scenes, you go off and you solve the problem. I used to love Agatha Christie and all of these novels, but this book really told you how you could become a detective. And so I was determined from age 11. I was passionate about this. I knew that this was what I wanted to do. And I think this is something that is actually a very good lesson for everybody. It's always good to have a passion to do something, okay? Because then you can have a good focus to your life. It doesn't matter what it is. Maybe you love video games. Maybe you say, oh, I'm going to make my life playing video games. Well, don't play them. Maybe what you want to do is write them, devise them, find some new ones, and make that into a career. And so I'm very, very keen always on following your passion. If you have something you love to do, try and make a career of it. And the great thing about it is if after 2 or 3 years you get passionate about something else, follow that. Don't think that because you've started in a particular path that you have to follow it. So, as I say, this teacher, Mr. Brooks at St. Stephen's School, really got me in love with puzzles. I go nowhere without a puzzle. I like Sudokus, I play chess, I like crossword puzzles, I play Go. I love games of this sort. But then I discovered chemistry. And I discovered chemistry because as a Christmas present, I was given one of these simple little chemistry sets. And this was in the days when chemistry sets really were chemistry sets. They were not these rather flaccid things that you find at the moment where you can't really do anything interesting with them. In those days, you actually could do interesting stuff with them. You could sort of mix things up and they'd turn yellow and red and so on. And I quickly went through all the experiments that were given with the chemistry set, and then I went down to the local library and started reading about chemistry. We didn't have those sorts of books at home. I grew up in rather a poor family, but I was not aware that I was poor. We had a great public library. I'd just go down there and get what I needed. And I got absolutely hooked on chemistry, and I discovered that I could make fireworks with the chemicals that were in this set that I'd been given for Christmas. And so from then on, it was fireworks, explosives. Oh, I just loved all this stuff, you know. Didn't do most of it at home. Most of it, we went down to the local canal, a couple of friends of mine, and we set off various things. But I really loved this and the chemistry and what you could do and how you could make the colors in fireworks and so on. And it's interesting because I've been doing a little informal survey among a lot of Nobel laureates in science, and more than half of them, this is how they got interested in science. And nowadays, you know, instead of selling chemistry sets where you can actually do something useful and have some fun and, you know, maybe risk blowing off a finger or two, the politicians have decided we can't do this anymore, right? They make these unbelievably mundane chemistry sets. They're absolutely hopeless. It's as though they really don't want young people to get interested in STEM subjects and find out about chemistry and the real world. So if I were a politician, this is one thing I would change. The first thing I would do to let kids get on and do things that are dangerous. You know, danger is a good thing. Almost everything worth doing in life is risky. I always think about skiing. You know, skiing is a great sport, but it's an accident waiting to happen. And you do it until you have your accident, and then you stop, and then you do something else. life is, almost everything in life really worth doing is a little bit risky. So this was Bath. This was the high school I went to. It's a City of Bath Boys Grammar School. So in those days, the kids who were considered to be a little bit smarter than the others were sent off to the grammar school. Kids who looked as though they might be very good at more technical things were sent off to a more technically oriented school, but not someone who was going to have an academic career of any sort. Okay? And so what happened at this school, and I got there, I can tell you why I got there. I got there because I got the highest score in mathematics of anybody of my year in Bath. I didn't find out this until many years later, but this was why they sent me there. But of course, by this time, I'd sort of gotten a little more interested in chemistry than I was in math, but I was still always good at math. So I increased my love of mathematics. We had a very good math teacher. Actually, we had a couple who were really good. So I loved that, and I learned to play the violin. I went to the school orchestra and so on. And that, for a while, was very nice, but when I left school and went to university, I applied to join the university orchestra. And I was told that unless you were a music major, you couldn't join their orchestra. I mean, it was appalling. And so for many years, I abandoned the violin altogether and only picked it up later when my oldest daughter got interested in doing something, and we did it together for a while. We also, in Bath, is very close to this limestone escarpment called the Mendips. Very nice caves, excellent caves there. And I love this because caving is also a form of detective work. You know, you go down and you can make discoveries. You go down, you see a little tube, and you think, oh, let's go through there and see what's there. And so I made a few discoveries underground on Mendip. And again, it's this idea that being the first to do something, being the first to find something, this for me has always been very exciting. I love this feeling when you discover something for the first time. It doesn't matter what it is. You know, maybe it's just a good tourist destination, but I like the idea of finding stuff. And I also discovered jazz, which I like very much, but more traditional jazz, the sort of St. Louis style of jazz. Bath became quite famous for that, and we have a festival every year, and that continues to this day. Now, for university, when it came time to go to university in England, you had to take these exams called A-levels, advanced levels. And you take this normally when you're 18. And so I took the exams and failed physics. I had never liked physics. The teacher was awful. And I just didn't understand a lot of what was going on. And so I tried a little bit, but I just didn't do well in physics. And so because of that, I couldn't go to university at 18 as I normally would have done. I had to stay at school for another year and take the exams again. And this time, I actually did a bit of work to try to get the physics right, and I managed just to scrape through. And at that point, it was not possible for me to go to Oxford or Cambridge, because there they would only take the very bright kids who were age 18 and so on. And plus, they actually turned out not to have the best chemistry departments in England. It turned out there were two universities, Sheffield and Southampton, both of whom had really good chemistry departments. And chemistry was what I wanted to do, and so I ended up going to Sheffield. And they accepted me, I got a state scholarship and so on, and it was all very good. And I loved the chemistry there. It was really a chance to be a detective, but a different sort of detective. And so in this case, you could do chemistry and find out about things. And we had a professor, W.D. Ollis, who was fantastic. And he taught in a method that is very different from the way that most everybody else has taught for years and years. And in fact, is now the method that he uses is now becoming thought of by educators as a good way to do things. And that is, instead of teaching you to remember stuff so you could just regurgitate it in an exam, he taught the principles of things, and he taught you why you needed to know this. And he presented chemistry as a puzzle, basically. And the exams, every exam that he had, would have questions of the form, well, you were given this compound, and you did this to it and got this result, and you did this to it and got this result. What is the compound? And so you couldn't sort of learn this ahead of time. You didn't know. And so you had to go through the business of logically working out what went on. And this turns out to be a great way to teach people. And for me, this was fantastic. I had a great time. And so when I finished as an undergraduate, I decided I was going to go and do research, and I chose to go and work in his lab. But in fact, what was interesting was it was a terrific choice, but not because of him. It was a terrific choice because in his lab he had a postdoc called Kazu Kurosawa from Japan, and he is probably the best teacher that I've come across in terms of really teaching you how to do stuff. He was one of these people who you'd go to him and say, well, you know, I have been told I've got to do this, and so he would say, oh, yes, and this is how you do it. But he'd explain exactly why you were doing what you were doing. And then when you did it, it would usually work, but you would know why you'd done it. And this is so important, especially when you're doing research, to know why you're doing the things you do, not just because you're following a recipe, but because you understand what is going on underneath. So he was fantastic, and that was a very lucky break in my life. Now, the University of Sheffield has had a bunch of Nobel laureates associated with it. Up here is one of the very first, Hans Krebs, is George Porter, is Harry Kroto, who came along just after me, sorry, this is Florey and this is Krebs. Here's me. I turned out to be the first student from Sheffield ever to win a Nobel Prize. These other people were teachers there. And so when they discovered that I'd won this prize, they made a great fuss of me, which was all very nice. There's actually a building now that they've named after me and so on. But they were a great university. I really have had very good interactions from the day I first went there until later. Now, Harry Kroto also was at Sheffield University, and he was the second student to win a Nobel Prize. He won it 3 years after I did, but he'd gone to Sheffield 3 years before me. I never actually knew him when we were in Sheffield, but nevertheless, he's a great storyteller. He's a wonderful lecturer, and so he always mentions the fact that, you know, there was this fellow Roberts who won a Nobel Prize, but, you know, he was 3 years younger than Harry, and so Harry taught him everything he knew. And so in recounting that story, I always tell everybody, well, but, you know, it took Harry another 3 years to get his Nobel Prize, so that's maybe telling you something. Anyway, we've had a lot of fun about this. Harry is a great guy. Unfortunately, he has ALS, and he's not in very good shape. That's Lou Gehrig's disease. He's not in very good shape at the moment, but has one of the most wonderful senses of humor even now. So when I was at Sheffield, my PhD thesis project, I was given a piece of wood from a tree in Brazil and asked to find out what was in it. And so this is what I did, and I was looking for some particular compound that my chemistry professor had predicted should exist as a metabolic intermediate in the making of some compounds called neoflavonoids. And many other students had been given pieces of wood from different trees, and no one had found this compound that he predicted should be there. And in my tree, it was there. And so he thought this was wonderful. He let me alone. I got every single result I needed for my PhD thesis after 1 year because of this guy, Kazu Kurosawa, who made sure I did all the right experiments. And so I ended up, because in England you have to do 3 years for a PhD, I had 2 years to really do whatever I wanted to do. And one of the things that I thought I would do would be to see whether I could isolate some of these intermediates in the synthesis of this neoflavonoid. And to do that, I learned some plant tissue culture, and I thought I could maybe do all. It was a total flop. It was a disaster. Not something that was good for me. But the other thing I did was I spent a lot of time in the library. And I went looking around the library, looking for interesting things to read. And I came across this book up here called The Thread of Life, An Introduction to Molecular Biology. This was a book written by John Kendrew, who also went on to win a Nobel Prize for the structure of myoglobin. Very interesting man. But he wrote this book that described the early days of molecular biology. And it was really written, he was a chemist, and it was really written from a chemical perspective, and it sounded fantastic. I mean, this molecular biology stuff, I just couldn't get enough of it. And so by the time I'd finished the book, I knew this was my new passion. I wanted to be a molecular biologist. And so I went and talked to my professor, Ollis, who immediately said, oh, there's no such thing as molecular biology, stick to chemistry. And it turned out almost everybody in the Department of Chemistry in Sheffield said the same thing. There's no such thing as molecular biology, it's just chemistry. Except for one guy, a guy called Mike Blackburn, who encouraged me to sort of follow this line. And so when it came time to look for a postdoc, I applied to labs that really would give me the opportunity to get into molecular biology and get away from chemistry. I applied to 6 labs. They all turned me down except for one. And the one person who accepted me was a guy called Strominger, Jack Strominger. That's this man here. He, at the time, was a professor in Wisconsin, and he had worked out the basic details by which bacteria make their cell walls. And one of the things he discovered along the way was that bacteria put amino acids in as a bridge to sort of cross-link the peptidoglycan that forms the basis of the cell wall, sort of hold it all in place. And I was given the task of trying to work out how these amino acids got in. It was known they used tRNAs, but not much else was known about it. So I got started on this project. I worked with a man called Tom Stewart, who was a postdoc who overlapped with me for 6 months. And the idea was I would just pick up his project and follow through. Well, I got to tell you, when I got to Harvard and switched fields, and I couldn't understand what half these people were talking about, because they didn't use chemical terms. They all used acronyms. You probably noticed this, anybody here who's a biologist. There are so many acronyms that if you're new to the field, you have no idea what people are talking about. And I think this is also something that I've learned along the way, that communication is incredibly important. That, whenever you're talking to people, whether they're in your field or out of your field, it's really good if you can make sure that whatever you talk about, they understand. And I have something that I always made my students go through, which I call the grandmother test, okay? Now, the grandmother test is a very simple one. The student has to convince their grandmother that the grandmother knows what they do. So you go through, you explain it in sufficiently simple terms that at the end of it, the grandmother gets it and can then go and tell all her friends about how great the grandson or granddaughter is, right? Because they've actually understood it all. And I think this is so important. I think we should all do this. Very, very important to learn how to communicate with people who are not specialists. Because it doesn't matter whether they're a specialist or not. You can use the same language to the specialist too, and they'll get it as well. So that was something that I liked very much. Now, I was given the task of sequencing this glycyl tRNA that was specialized for working in bacterial cell walls. And at the time, this was a time when there were only a couple of methods available to sequence RNA. It was still very young. And one guy in Wisconsin, a guy called Bob Holly, had developed one method of doing it, which was unbelievably tedious. I mean, it really was tedious, but because Tom Stewart, who had started the project, had come from Wisconsin to Harvard, he was following this method. Well, after he went, I started to read about it and discover there was a much better method that was being performed by Fred Sanger in Cambridge, at the MRC lab in Cambridge. And so the first thing I did was to say, well, I'm going to abandon this method that Holly had come up with, and I would get going on this Sanger method. And to do that, you had to make very highly labeled, P32 labeled tRNAs. And this was not so easy because I was working on one from a Staphylococcus, and it just was very difficult to get enough of the labeled tRNA in order to then go and start doing all the methods. And to do that, I had to do a 500 millicurie prep of tRNA. So I took some Staph aureus, Staph epidermidis rather, grew it up in a vat and with 500 millicuries of P32. 500 millicuries of P32 is quite radioactive. At Harvard, there was sort of one wing over here and another wing few hundred feet away, and you could take a Geiger counter and detect it from one wing to the other. So this was kind of fun, you know. This was a sort of risk that the regulators don't like you to take. Anyway, so we couldn't do it at Harvard. They didn't have the appropriate facilities, so I went down to the hot lab at MIT and did the work down there to make this tRNA. And then when it had gotten down to a reasonable level, such that Harvard thought it was okay to have around, we took it into the cold room and finished off the purification. And the very first batch, the fraction collector stuck overnight, and everything went onto the floor of the fraction collector, okay? So this was pretty bad, but Jack was kind of decent about it, you know. He said, oh, this will be okay, we'll just get another batch of, and this was an expensive experiment. Anyway, so we got another batch. Second time, I sat up all night and watched this bloody fraction collector, you know, to make sure that we didn't have the same problem. So made the batch, took it over to Fred Sanger's lab, spent 6 weeks over there, and those were incredibly rewarding weeks for many reasons. So in Fred Sanger's lab, he had developed protein sequencing techniques and won the Nobel Prize for that already. He shared the DNA sequencing prize with Wally Gilbert. He could equally have had a prize for sequencing RNA. This is a man who, when I first met him, I thought he was the janitor. He is one of the most low-key people you could imagine. Just unbelievable, and yet a tremendous talent, you know. He was just fantastic at coming up with ideas and then telling you, well, you know, maybe it would work, and very modest about everything. But for me, he was really a model scientist. I think this is how all science should be done. He was a great guy. Unfortunately, Wally Gilbert, who shared the prize with him, is a very different kettle of fish. And what happened is that Wally came up with a different method of DNA sequencing, and he came up with a very detailed protocol of how to do it. And Fred never came up with a protocol. And the bottom line was that Wally's method just spread everywhere. And it was, however you look at it, it was demonstrably inferior to Fred Sanger's method. And so I always think that, you know, Wally, if anything, set the field back. You know, he set DNA sequencing back. It was probably 2 years before Fred's method really took up where it should have gone. And one of the saddest parts about that is that if Wally had not shared the prize with Fred, Wally could have gotten it for something else. He'd discovered the LAC operator and worked out how the LAC repressor operator interactions worked. Could have shared that prize with Mark Potashny, who did the same thing for Lambda. But the Nobel Committee, they really don't like to give the prize to the same person twice if they can avoid it. And so I always say that, you know, it was unfortunate that because of this, Mark Potashny never got his prize. But, you know, the Nobel Prize, unfortunately, is full of stories like that of people who should have got it and didn't. So went to Fred's lab, learned how to do it, learned how to sequence, and came to the attention of Jim Watson. I actually got to know Mark Potashny very well because I taught him how to sequence using the Sanger methods, and also Tom Maniatis and a number of people. I was the first person in the Boston area to be doing this. I came to the attention of Jim Watson. And so one day I'm in a seminar, and Mark Potashny comes up to me and he says, you know, Jim Watson is looking for someone who will sequence SV40 DNA down at Cold Spring Harbor, and he's going to come up and talk to you about this. So I said, fine. So I wait, and I wait, and I wait, and no sign of Jim. And then Mark comes up to me about 2 or 3 weeks later in another seminar and says, you know, Jim doesn't actually know who you are, so maybe you could go and introduce yourself. So I proceeded to do that. Went down to his office at 5 o'clock at night, 5:30 or something. Knocked on the door, went in, and Jim says to me, you know, I say, I'm Rich Roberts, and he says, oh, yeah, Mark told me about you. He says, I want you to come down to Cold Spring Harbor and sequence SV40. And the whole thing lasted maybe 5-10 minutes. And at the end of it, I'm just walking out of the door, and he says, you know, you probably should come down to Cold Spring Harbor and give a little talk about what you're doing. But don't worry, he said, the job is yours. So, you know, this short interview, I got this job at Cold Spring Harbor, so I did go down to Cold Spring Harbor and did get the job, and I spent 20 years down there. I was fairly happy for most of those years until I became assistant director. because prior to that I could do research, and then my research kind of dropped off at that point. But I had a very good time. However, most of the time I was fighting with Jim Watson. And the reason is that when I went down there, I'd been hired to sequence SV40, but I get down there, I discover there are 2 more groups already sequencing SV40. So, why would a third one want to do it? And so I thought, well, what I'll do is try to do something that will sound as though I'm interested in sequencing SV40, but in fact, it is rather different. And that was to develop methods for sequencing DNA. Because what had happened is that the first type 2 restriction enzymes had been discovered by Ham Smith, and it was clear that here were some enzymes that would take DNA and cut it up into small pieces. Now, the reason that RNA sequencing was developed before DNA sequencing is that whenever you're going to produce new methods and try to get new methodology going, you need small things to practice on. Okay, you can't practice on something that is so big that you have no alternative way of checking whether your answer is correct or not. And in RNA, there were nice short RNA molecules. You could do it by several different methods and know that you were getting the right answer. With DNA, there were no small DNA molecules. And so I thought these restriction enzymes were going to give us the opportunity to make the small DNAs that we could use to build methods for sequencing DNA. And so one of the things I did when I got down to Cold Spring Harbor, I purified the ones that were known. There were 5 known at the time. And then during the course of purifying one of those, we discovered there were actually 2 enzymes in there, not just one. And I brought down with me from Jack's lab a whole bunch of strains because I thought I might end up doing a little work on the tRNAs involved in cell wall biosynthesis. And so I brought about 20 strains from Jack's lab with me in case I wanted to pursue that. And so we started to look there and see, well, do these strains have restriction enzymes in them? And lo and behold, everywhere we looked, every strain we opened up, except for just very few, had a restriction enzyme, and they were all new. They all recognized different sequences. And so it occurred to me that maybe we could use the specificity of the restriction enzymes in order to work out what DNA sequences were. And so I went full head into that, to which Jim Watson immediately came and told me I was wasting my time, that I was hired to sequence SV40, and why didn't I get on and do it? And so we had a few fights about that. But I was convinced that these restriction enzymes were going to be useful, not just for sequencing DNA, but more generally, for allowing you to make small pieces of DNA that carried genes. You could use them for mapping. There was just a million things that could be done. And of course, these later led to be some of the major tools in recombinant DNA. And so without these enzymes, the biotech industry would have been much slowed in its development. Everybody was using these. And so in the course of this, what happened was that I went to Jim and I said, look, at the time we had 30 enzymes. They were all different, all recognized different sequences. I said, let's start a company. Let's sell these enzymes to the community instead of giving them away, which was what I was doing. Make some profits and feed the profits into Cold Spring Harbor, and let's support the research at Cold Spring Harbor in this way. To which Jim, in typical fashion, said, no, you're crazy, don't want to do this, it's very dirty to be associated with industry. And plus, you can't make money on doing this, and he was not interested. And yet here I am, we're giving all our stuff away. And so I started looking around to see if there was someone else who would be interested in this, and ran into a fellow called Don Combe, who had started New England Biolabs. He invited me to go up. I went up and discovered that New England Biolabs at the time was a basement in Beverly underneath a hairdresser's. There was Don Combe, his wife, and one technician, and they were operating on little makeshift benches that they'd put together, and they had an old centrifuge that they'd bought as a spare from Harvard. Harvard sell off their old stuff. And this was where they were starting to make these things. And in 1974, the company got started. It was incorporated in 75, and they made $200,000 selling restriction enzymes in 1975. And this was a lot of money, and from just a very small makeshift operation. And so I always say, if you're going to put up an incubator, everybody likes to incubate companies. Okay? You don't want to put up these fancy buildings. You want to find basements they can go into and find garages. You know, you look at Bill Gates and Microsoft and Steve Jobs and, you know, these people who get these companies going, they do it on the cheap. They don't do it by going into these fancy incubators. And so I think there's another lesson there that's worth doing. So anyway, so that was sort of the story of restriction enzymes. The Nobel Prize for this was given in 1978 to Dan Nathans, Ham Smith, and Werner Aber. Werner Aber discovered the very first one, but it turned out it wasn't very interesting. Ham Smith discovered Hindi2. He called it endonuclease R. It was actually a mixture of two enzymes, which I found as soon as we started to try and purify it. And this was the enzyme that made all the difference because it gave specific fragments of DNA. And Ham only was interested in it because he thought it was promoting recombination in Haemophilus, had no idea what it might be useful for. But fortunately, Dan Nathans, who shared the next lab to Ham, figured out what it was useful for and started using it to map SV40, and the rest of it is history in terms of mapping and so on. And so these three were just a tremendous trio to win this prize. It was fantastic. Let's see where we're going now. Okay, so this is a summary of the things I did while I was at Cold Spring Harbor. So in the first 2 or 3 years I was there, we found 30 restriction enzymes, all but one of which was different. In 75, I became scientific advisor to New England Biolabs. They'd actually offered me a partnership in the company, but I had no interest in that. I, was just a new investigator down at Cold Spring Harbor. I wanted to do research. I still had my Nobel Prize winning work to do, so I couldn't leave and go to a company. I produced the first restriction enzyme list, which was a list of all the known enzymes that we circulated widely. This became the precursor of a database that I now run and have run. It was one of the very first molecular biology databases. We found many RM system genes. We did a bunch of cloning. And by 1978, we'd started doing bioinformatics related to restriction enzymes at first, but later related to DNA sequences and RNA and so on. But of course, during this time, Jim was constantly telling me why was I wasting my time working on these restriction enzymes and so on. And eventually it got to be so wearing that I thought, well, maybe we'll try another project too. And so I got a postdoc to come down, Richard Gelinas. I don't have his picture yet. He came down, and we decided that what we would do would be to try to map a promoter on a eukaryotic virus, something that would resemble a human promoter or a mouse promoter or some higher organism's promoter, and see if the sequences at the 5' end, which were going to drive the expression of a gene, were the same in eukaryotes and in prokaryotes. We already knew what they were in prokaryotes, thanks to work that had been done at Harvard with Wally Gilbert, Jim Watson's lab, Mark Potashny's lab, and so on. And so we knew more or less what was going on there, but we had no idea about what was going on in adenovirus, which was the virus we chose to work on. And so we had a very, very simple idea. When RNA is first made, the 5' end has a triphosphate on it. So it starts with ATP or GTP, usually ATP, and it has a triphosphate on it. Runs along, you add one base at a time with the RNA polymerase until you get to the 3' end, and then the 3' end has poly A on it. And so here you've got molecules that if you cut these at all the G residues, which you could do with this enzyme, a nuclease called ribonuclease T1, you would make a series of oligonucleotides. And there would be basically just 3 kinds of oligonucleotides produced. One from the interior part, which had a 5' phosphate and a 3' hydroxyl. One from the 3' end, which would have a big poly A tail on it. And so you could actually get rid of those very easily, just run them over an oligo DT column. It would catch them. You could get rid of those. But what you would have would be something with a triphosphate on it. And if we could find the oligos that had the triphosphate, these would tell us where the RNA started, and then we could sequence upstream of that on the DNA and work out what a promoter was. Very simple idea, very simple experiment. And so Richard got started doing this, And we started with adenovirus because quite a lot was already known about adenovirus in terms of transcription and genes and where they were. And we knew that early in infection, there were RNA transcripts made coming in from the ends of the virus. Adeno is a linear virus, so there were transcripts coming in from the end. And so we started trying to find those transcripts, but they were present in such low quantities, we could never get enough of them. And then, while we were doing that, it was discovered that RNAs made in adenovirus and in other systems had triphosphate caps on them. They had a 7-methyl G, 2'-O-methyl G, and then a G hooked up in a 3'5" configuration. They had a different structure, but what this meant was that they effectively had 2 3' ends because the G that was on the cap was facing in the wrong direction. And so it had a 3' dihydroxyl group. Poly A had that too. And that actually gave us the hook so that we could separate the capped oligonucleotide, the one from the very end, from everything else. And I came up with a method for doing this that was based upon boron chemistry. Boron turns out to be very nice for binding things that have cis diols, that have 2 hydroxyl groups on them. Came up with this method, and so Richard did the experiment, and we were expecting, because we knew there were about 20 different messages, that we would see 20 different oligonucleotides corresponding to the start points of all the messenger RNAs. We did the experiment, and he only found one capped oligonucleotide from these 20 messages. Well, it didn't make sense. This was not what we were expecting. And so, I had a little talk with him and said, why don't you go back and be a little more careful next time and do the experiment properly? So he did. He went back and did it again and got the same result. So we had another talk, and he went and did it again and got the same result. And so I said, Richard, why don't you sit down, let me do it, I'll show you how to do it properly. And so I went and did the experiment myself, and sure enough, I got the same result. And at that point, of course, I believed it, right? So, one of the nice things is when experiments fail in this kind of situation, it's one of the greatest things that can happen. Failure is a great thing because it's telling you one of two things. Either you screwed up, maybe you dropped the sample on the floor or just didn't lay it up properly, but maybe nature is trying to tell you something. And in this case, nature was trying to tell us something very big. And so I always love it when students come in and say, you suggested I do this experiment and it didn't work. Great. Maybe there's a discovery here. So you don't ever be disheartened by failure. Failure is a good thing. All the great discoveries are usually made by people doing an experiment and it didn't work. So Richard did the experiment. It took us a while before we could figure out exactly what was going on. So we could see that this one oligonucleotide at the end of the message came from every individual messages. If we try to purify individual messages by hybridization, they all had this same structure at the end. Now I can tell you, most of the people at Cold Spring Harbor whom we told this result to said, it's an artifact. Okay, you're just not pulling out what you think you're pulling out. It's something that's very odd. But we knew that we were really doing the experiments correctly. And we just went on, and we got into this mode. Every week, we'd have a little autopsy on a Saturday morning to understand why last week's experiments hadn't worked and why next week, maybe we could do the biggest and best experiment next week. And so on one of these Saturday mornings in early March in 1977, we're sitting having our Saturday morning autopsy, and Richard is there, and he's proposing some unbelievably complicated experiment. It was I just wasn't paying attention. And then suddenly, out of the blue, the experiment to do came to me. And so I say to Richard, sit down, here's what we should do. And so the experiment was it was an electron microscopy experiment. But neither Richard nor I were electron microscopists, but we were lucky, because just down the hall were two excellent microscopists, Tom Broker and Louise Chow. And so we went down to them and said, we've had this idea for an experiment. If we make the reagents for you, will you do it for us? And so they looked and they said, well, it's never been done. No one's ever quite done this thing before, but sure, it should work, and we'll do it. And so Richard made the appropriate substrates that we needed for the experiment over the weekend. And on Tuesday morning, Louise Chow spread the results on the electron microscope, looked at them, and lo and behold, splicing was discovered. Exactly what we had predicted should happen, had happened. It wasn't quite so straightforward as that. The experiment was based on a false premise, but it didn't matter because the fragments that we chose to look at worked anyway. The experiment is sort of illustrated up here. I don't really want to try and go into it in too much detail, but the idea of the experiment was if you can take a messenger RNA, you can hybridize it to DNA, and you get a very nice hybrid. Now, if there are sequences that do not hybridize to the DNA, that they will stick out. They'll be sticking out. Just the poly A is not coded in the DNA. It just sticks out because it's added post-transcriptionally. And you can detect it by putting in oligo-DT, and then you get a nice signal in the electron microscope, get something nice and visual. We were interested in the other end. We wanted to know what went on at the 5' end. We thought we knew where the structure might be coded, and so that was the probe that we used. And it turned out that it was the right probe, but it was for the wrong reason. It was the wrong sequence. But it didn't matter because the right sequences were on that probe. And at this point, splicing was discovered. We had a picture, and everybody believed us. You know, we'd had almost a year's worth of biochemistry before that. And I remember, you know, I went up to Jim's office and told him all I told everybody who would listen about these experiments, and almost nobody would believe them. One of the few people who really believed them was a man called Georgy Georgiev, who was head of the Molecular Biology Institute in Moscow. And he was an instant convert. He thought this was great. But most other people just didn't believe it until they saw this electron microscopic picture. And so so we got the results, 10, 11 o'clock in the morning. By the end of the day, everybody was talking about Nobel Prizes because this was a huge discovery. It overthrew, as you heard earlier, all of the existing paradigms about how RNAs were made and what genes look like in higher organisms opposed to lower organisms. And this diagram tries to explain it a little bit. So basically, the gene is divided in a eukaryote. It spreads over a huge distance. And there are little bits of it that code for the protein, but they're separated, and they sit on some sequences that we call exons. And then in between, there's a region that just never codes for anything that we know about in terms of proteins for the most part, and that's called an intron. These names were invented by Wally Gilbert. And we now know that what goes on is that you make an RNA that copies the whole thing, and then you treat it as though it were a movie. You know, you just saw all the Oscars, and they were giving out Oscars for people who were busy editing transcripts, film transcripts, that is. That's what happens here. You basically make the whole RNA, and you take it to the cutting block, and you cut and splice and cut and splice. You take the bits that make sense, join them together. The rest of them you throw away in just what happens when you make a movie, so that eventually you get a movie that looks as though it was just shot continuously all the way through because of the good editing job that's done, and that's what happens in higher organisms. This is how we make the RNAs that are ultimately going to code for all our proteins. In bacteria, you don't do that. Just make a direct copy of the DNA, and then you've got what you want. So it was really a very, very nice discovery to be made because no one had predicted it. You know, very often you make a discovery and all you're doing is confirming someone's hypothesis. This was not hypothesis-driven science. This was science that we were just looking to see how something was made, and at almost every stage our hypothesis was shown to be incorrect. So it was nice. So this was good. And of course, you know, one of the disappointing things here, which I think points out one of the problems we have in science today, and certainly had back then, is that as soon as we made the discovery, everybody went back, looked at their own data, and discovered they had the data in the notebooks that showed in some other system exactly the same thing was going on. I went to a meeting at Cold Spring Harbor in which the RNA virologists were there, and Mike Bishop stands up, who later went on to win a Nobel Prize for the retrovirus work, showed this slide in which he clearly had all the data showing that splicing was going on, and so he was able to explain it in terms of splicing. At least he didn't try to claim he discovered it, but an awful lot of people at the time did try to claim they had discovered it. And that was kind of disappointing that something like that goes on. Now, the first thing we did after this, you make a big discovery, you want to capitalize on it. What can you do? Well, most of the stuff that really needed doing was all electron microscopy. And of course, as I said, neither Richard nor I were electron microscopists. And so Tom Brocher and Louise Chow were able to go on and produce just beautiful work showing what was going on using electron microscopy to work out where all the messages were coming from. Richard and I decided we would try to do something in vitro. Maybe we could work out the biochemistry of this process by doing it in vitro. And we set up a system and started to do some work, but we picked the wrong RNA polymerase in order to make the RNA in vitro. And Tom Maniatis at Harvard picked the right RNA polymerase, and so he was able to make hay on in vitro splicing. Set it all up, discovered lariat, just discovered a huge amount of stuff about how to do it, was immensely successful, and we got nowhere. So for us, this was a great disappointment. So I sort of settled back and did more of the restriction enzyme work, which has really been a love of mine for the whole of my life. But one of the things that we did do at this time was to realize that if, in fact, there were signals in the DNA that would be necessary to tell the organism where to do this splicing process. Maybe by looking at sequences, we could find them. And so one of the things we got very interested in was using computer programs to look at DNA and RNA sequences. This was a time before there was such a term as bioinformatics. Nobody knew about anything of that. And there were no programs to be doing this sort of stuff. And so we ended up writing a lot of software. I ended up teaching myself how to program because this was really something that was very new, and the traditional programmers, what we needed to do was really trivial stuff, and they were only interested in the more difficult stuff. And about that time, one of the people that I interacted with is a man called Carter Burwell, who is now, he writes musical scores. He was a programmer at Cold Spring Harbor. And essentially, every time I had problems when I was learning how to write programs, I would go to him and he'd put me straight and tell me what to do. He was actually up for an Oscar on Sunday night. He didn't win, unfortunately. There were a lot of us from Cold Spring Harbor really rooting for him. He was just a great guy. He wrote the musical score for Carol, if any of you have seen that movie. Anyway, he's really a great guy. So I owe him a gratitude of debt too, because one of the things that I love is computer programming, computer programs, bioinformatics, and a lot of what I do these days is bioinformatics. So this is Richard Gelinas, the guy who did the bulk of the experimental work on the biochemical side of splicing before Tom and Louise came along. He is now working at the Institute for Systems Biology that Lee Hood set up in Seattle. This was the first paper that we put out on this, showing about this long oligo that was present on all of the messenger RNAs. Okay. And then this was the discovery of RNA splicing. We called the paper, we had a lot of discussion about how we should title this paper, and in particular, the word amazing. You can think of many words that you could put here. And so I liked amazing. And so we sent it in, and 3 referees all wrote back and said amazing was an inappropriate word to use in the title of a paper. And Ben Lewin was the editor, and he wrote a letter and said, if we could change this word and do 1 or 2 other minor revisions, we'd be okay. And so I called him up and started talking, and during the conversation I said, when you first heard about this, what did you think? And he said, oh, it's amazing. I said, I rest my case. And so by a little bit of trickery, I convinced him this was okay. So apparently this is the first time that the word amazing was ever used in the title of a scientific paper. So I'm kind of proud of that too. So this is New England Biolabs. When I moved there were just 3 people working there. In 1992, after I'd been at Cold Spring Harbor for 20 years and had become assistant director and kind of got bored with the administrative work that was involved there, I decided I was going to move. I thought I might set up a sequencing company. This was at the time when people got interested in sort of high throughput sequencing, and there were machines being made and so on. And then New England Biolabs heard that I might be interested in doing something on the commercial side rather than just pure research. And so they made me an offer which I felt I couldn't refuse, which was basically they said, you know, come up, be a research director, and you can do whatever you want on the research front, which sounded pretty good to me. So that's what I did. I moved up there. And 1 year later, of course, I won the Nobel Prize. Oh, before I do that, so this was the second building after we moved out of this basement under the hairdresser. This was the first building that we put up. And then this is the current building that we put up in 2005. We've been there about 10 years now. It's a beautiful building. If any of you get the chance to come visit Biolabs, you should certainly take it. It's almost an art gallery inside. So Don Combe is very keen on art. He just loves art, and we have a lot of very interesting art inside. And we do a lot of research there. Now, since moving to Biolabs, there's a bunch of things that I've done. Got involved in cloning restriction enzymes, not really doing a lot myself, but finding things, exploring the limits of bioinformatics, and this is something that I'm really very hot on. I'm keen on bioinformatics at the moment, and most of what I do these days is on a computer, although I have a small wet lab that follows through on things I suggest they should do. And then we decided that when we were trying to clone all the genes for our restriction enzymes, that it turned out it was not that easy. Restriction enzymes are a bit difficult to clone, and so we thought the thing to do was actually to just sequence the genomes for these things. This was when 454 sequencing came along. And so for the last 50 restriction enzyme genes we wanted to clone, we sequenced them first, used bioinformatics to find out where the methylases were, and then looked around to see if we could find the restriction enzyme genes. And 48 out of the 50 we were looking for, we were successful in cloning using that method. The reason I'm so keen on bioinformatics is that if you look around, what you see is there's an enormous amount of sequencing going on. And in fact, it's clear that ultimately we're only going to know about the biology on this planet by sequencing everything. But if you can't interpret that sequence, what's the point? Why do it? And so we really face a big challenge. We've got to understand how to interpret these sequences properly. And at the moment, there's a tremendous amount of money being spent on sequencing, and there is much less money being spent on actually trying to work out what the genes are doing. I'll give you an example, Mycobacterium tuberculosis. You know, tuberculosis, nasty disease. We know what causes it. We have sequenced hundreds of isolates of Mycobacterium tuberculosis. How many of the genes have we actually tried to work out what they do since the first sequence was done? The answer is about a dozen. Okay? It is pathetic. It's absolutely, here is a major killer, a real problem, and we don't know very much about the genome and what the genes are doing and how they all work. And we have to do more of this. I think that there are some serious problems. So this was the prize with Phil Sharp and myself in 1993. I have to tell you, I can tell you more later if you want to know, but the Swedes throw the most magnificent party. It is just unbelievable what they do. You arrive at the end, well, you get on the plane to go over there, and the first thing they do is announce they have Nobel Prize winners on board. They provide everybody with champagne, and they all toast you. You know, and this is while you're still on the way. This was at Newark Airport, pretty much. You get there, they give you an attaché, and you go through for about 10 days a series of events in which, if any one of these events at lunchtime or the evening had been the only thing that happened to you during the course of that year, this would be the most memorable thing that happened during the year. It just goes on and on and on. It's fun. I recommend it to everybody. So one of the things that we did just before going to Sweden you know, you have to give a Nobel lecture, so you've got to get there and you have to tell everybody about all these wonderful things that you've done since you made the discovery. Well, as I say, we didn't do very much. We sequenced adenovirus, that was about it, but we really didn't do all that much, and my research had switched completely. And we were actually interested in HHA1, a DNA methylase, and we had discovered about 3 months before I had to go to Stockholm that the way in which this protein interacts with DNA is the base that it's going to methylate, it flips it out of the helix, 180 degrees, right out of the helix, so goes into the active side of the protein so the chemistry can be done. And if you imagine, you know, once you've got all these bases in a helix, it's quite difficult to do chemistry on them. And so we had made this discovery, and I was able, as my Nobel lecture, to talk about this discovery. You know, and if you make a discovery, there's no better place to talk about it than in Stockholm. I mean, this is a lesson worth learning here. So anyway, so this was very good, and we predicted that many enzymes that did chemistry would do this. We now know that this is true. Almost every enzyme that does chemistry on DNA uses this same mechanism. And not only that, if you look at this, you can see how easy it would be to open up the helix once you flip one of the bases out. And we now know that RNA polymerase, when it is first starting to transcribe DNA, it flips out, in the case of E. coli, base -11 to the start point of transcription. This is how it begins opening up the helix. We think the same thing probably happens for DNA replication too. So this was a very nice discovery. It was something good to be able to talk about in Stockholm. Now, as I said right from the start, I've always loved puzzles and games and so on. I spent my Nobel Prize money putting a local croquet lawn in front of my house, at great expense, I might add. I think even to this day, my wife doesn't know quite how much it cost. But it was very expensive, and the Scientific American ran a little article explaining how people spent their Nobel Prize money. And they were talking about people like Gunther Blobel, who gave his money to help rebuild Dresden. And they quoted me as someone who spent it in a somewhat less good way. And sort of essentially they hinted that I'd wasted the money by putting a croquet lawn in front of my house. In fact, I have had so much enjoyment out of this, it was worth every penny. But without the prize, I could never have afforded it. So that was very nice. I became the stud muffin of science. One year in the 1997 calendar, someone came and approached me and said, would I do this? You know, most of them were these young guys. They had hairy chests and looked good, and the ladies liked them. And when my wife heard about this and heard that I'd said yes and the photographer was coming, she left the house and said she didn't want to have anything to do with it. But later on, she decided it was actually a little more tasteful than she had imagined. So that was very good. A few other things that have happened in whenever it was, 2005, they named this building after me at Sheffield. In 2008, I was knighted by Prince Charles. The ceremony is shown up here. This is actually a very nice ceremony. You sort of go into Buckingham Palace and you have to go and they sort of prime you as to what you're supposed to do. It's all incredibly formal. But my wife and kids loved it. They thought this was great. I thought it was sort of okay, but they just loved it. And so that was very nice. And one of the things that I'm rather proud of is that I've really helped to organize a lot of the Nobel laureates to do good things. And this is because one of the things I find as a Nobel laureate is that people come, they ask for your opinion on things, irrespective of whether you actually know anything about it or not. But anyway, they come and ask you your opinion. And they listen. I've been amazed at how many people listen. And so I thought, well, you know, here is an opportunity for the Nobel laureates really to try and do some good. And so here's a few things that I've gotten involved in organizing. One was the Open Access Publication Initiative. This was something that I got quite a lot of laureates, 49 science laureates, to sign on to a petition that we sent off to publishers to try and get open access going. In 2006, there was an issue in the Benghazi Children's Hospital where Muammar Gaddafi had accused 5 Bulgarian nurses and 1 Palestinian student of spreading HIV to a little over 400 children. And it was clear as things developed that they hadn't done this. It was just poor medical practice. In Libya, maybe even today, they thought sharing needles was a good thing. So, you get a vaccine, just go and vaccinate everybody. And they spread the virus in this manner. And I organized a number of laureates. We got 119 laureates to sign a letter. I went to the Libyan embassy for the United Nations down in New York, talked to the ambassador. I eventually ended up going over to Libya and meeting with Gaddafi's son, and we got the nurses out, basically. And so that was an interesting thing and something worth doing. Daw Aung San Suu Kyi is the Peace Prize winner who was in house arrest in Burma for a long time. We got 219 laureates to sign on to that, and we tried to do what we could. We were not successful in that one. Liu Xiaobo is a Chinese Peace Prize laureate. He is in jail in China at the moment, and we've been trying to do something there. Camp Ashraf is another thing I've been involved in. This is a bunch of Iranians who are in Iraq and are constantly being harassed. The CIRCB in Cameroon was a research institute working on AIDS. And in this particular case, one of our laureates, who will remain nameless for the purposes of this talk, really did some very bad things, and he was about to become director of this institute. We got him thrown out. And at the moment, I'm very much starting a campaign pro-GMOs. What has been going on with genetically modified organisms and the Greenpeace and the other parties who are against these and trying to get GMOs and pretend that they're dangerous when they're clearly not is just getting totally out of control. And so I so far have 75 laureates who've signed on to a letter that we've written. We're going to launch a big campaign before too long. I have a website I'm putting together where people can join us. I hope some of you will think that's worthwhile. But this is anti-science of the very worst kind, and I think unless we can get something done about it, the developing world are going to suffer tremendously. We don't need GMOs. We're fine. We have plenty of food. But in the developing world, if they don't get access to this technology, they're going to be dead, and that isn't a good thing. So, just want to finish, talk about the importance of luck. Okay, it's almost time. Let's see how quickly I can go through this. So I learned my first lesson about luck playing billiards. I was a very good billiards player. In fact, I almost lost a year at school by playing billiards and snooker instead of going to school. And I went to audition, actually, to become a professional billiards player, because I was pretty good. But anyway, that didn't happen. That's another story. But there was an old gentleman one time who was watching me play, and I'd had a lucky shot, and I just fluffed the next shot. And he came up to me afterwards and he said, the importance of luck is that everybody has it, and when you have it, if you don't take advantage of it, you're a fool. Okay? Because most people, when they have luck, they will take advantage of it. And so what you have to do when a lucky break comes your way, concentrate even harder on the next shot or the next point in your career or whatever you're doing. Everybody has luck, but you don't want to throw it away. You have to make the most of it. And I think that is really valuable advice. Kazu Kurosawa was the Japanese postdoc I told you about. I ended up at Harvard instead of going to Wisconsin, which is where I thought I was going because Jack moved just a couple of months before I had to go. I'm looking for eukaryotic promoters in adenovirus. Well, we could have chosen 100 projects. We chose this one. It was lucky. Here's one that you probably haven't heard about, taking an early plane. You all know what happened in New York on 9/11. There were 2 planes that went into the Twin Towers. The first of those planes I was booked on. About a week, 10 days before that happened, the meeting I was going to in California was canceled and moved a day earlier. And so I changed my reservation about 10 days beforehand, and I flew over to California one day before that happened. But for that change in meeting, I would have been on that plane and I wouldn't be talking to you today. So luck is unbelievably important, and everybody has it. You just can't believe how important it is in your life until if you go back and start looking, you'll find there are a lot of lucky things that have happened to you. And finally, studying bacterial RM systems has just given me such a wealth of fun, enjoyment, but also had a big impact on biology, on the biotech industry, got me my current job, and it is fun. And I will talk to you, for those of you who want to come to the talk later, I will talk about what I'm currently doing with RM systems, which I find incredibly exciting. So I see the time has almost gone. Well, let me just put up this last slide. Is about humans and bacteria. You know, we now know the metagenome is incredibly important. The future of medicine is going to be heavily influenced as we learn more about the metagenome. And there are an awful lot of bacteria that live with us, a lot of viruses that live with the bacteria. We know almost nothing about these. And so I think here's a great area for research, and one that is going to have a major impact on medicine. So I thank you for all your attention, and if you have any questions, I'd be happy to answer them. Thank you. I think there is a microphone circulating somewhere if someone has a question.
Just out of curiosity, what programs do you use, or languages?
Well, I don't write anymore. I mean, I can tell you a little anecdote. So I learned Fortran. Right? So this was back in the late 1970s. And what happened is that one night I was debugging a program, finished it, it worked, looked at the clock, it was 8 o'clock in the morning, and I hadn't realized I'd worked all night. And the same thing happened to me about 3 days later, and I said, this is it. You know, I am so hooked on this, this is going to be the death of me. And so I stopped programming, and now I have other people doing it. And so all the modern programs are what the programmers who work for me now use. So C, Perl scripts, and a variety of modern programs that are useful. But I try to stay away from that, having already had this experience of almost overdosing on it.
I read Crick's autobiography with great interest, and toward the end he said humans will become extinct, but we are animals and we will become extinct, but we will live on in another form. Mm-hmm. Can you elaborate any more on that?
Well, you know, I can't,
no.
but I can tell you what I think or thought about Francis Crick. He was the brightest human being I'd ever met. I mean, he was one of these people I met first when I was a postdoc at Harvard, and I was explaining to him what I was doing, and he mapped out about 20 years of research for me during the course of a half-hour conversation. He was incredible, and I ran into him many times after that, and just an incredibly bright, smart human being. So maybe he had some insights that I don't have, but he's one of my great heroes. He and Sidney Brenner, very, very smart people.
So in your talk, you mentioned several influential people, and I'm curious, how can you determine whether someone is going to be a fruitful collaborator or is going to be getting your research moving forward? How can you, or do you have to determine that, or you just use
luck? I mean, I think what happens is that as you meet people, you pretty soon get an idea of who is good and who is not good. Richard Gelinas, for instance, worked as a graduate student in Fotis Kafados' lab, and I knew that experimentally he was a very good experimentalist. And this was what was needed for this kind of project. And what I typically look for is people who are enthusiastic about what they're doing. So if someone comes and interviews with me for a job or as a student or whatever, if they are really passionate about what they want to do, I will take them almost irrespective of what their previous educational background is. I don't think early education necessarily tells you very much about how good someone is going to be in the long run. I mean, if you looked at my educational background, you probably wouldn't hire me. So.
What do you consider the most underrated and overrated discovery of maybe the past decade in science?
Gosh. Well, one of the big discoveries is obviously CRISPR, and the fact that you can now do genome editing with it. It is quite powerful. Will that be the best way of doing genome editing? I don't know. This came also from studying bacterial immune systems. Bacteria do the most amazing things. Probably the most underrated discovery, I wouldn't say over the last 10 years, but the fact that the discovery that DNA was the genetic material, which was made by McCarthy and McLeod, they never got the Nobel Prize for this, and they should have done. I mean, this was an absolutely monumental discovery, which everything else that followed on DNA really depended upon very heavily. In recent years, I don't know. It's really hard to know. I don't have I can think of a few things that I don't think were so great that have been lauded, but on the whole, I don't want to go into that.
You talked about the importance of luck. What about intuition in your life? Has that played a part for you?
Well, intuition is a very interesting thing. I don't know if you've read Daniel Kahneman's book. So he has a book called Thinking Fast and Slow. Kahneman was a psychologist who won the Economics Prize. He had this book called Thinking Fast and Slow, and he talks about the ways in which we think about things. And the things that most people, and certainly most ladies, think of as intuition is sort of this fast track that somebody asks you a question or you see some facts and you immediately think you can explain them. And this is sort of the intuitive thing. It's the way that the brain typically reacts to things. But then sometimes you have to look back and you can't immediately say something. Then you have to really sit and think and analyze it. And a lot of people who use intuition don't go back and do the analytical stuff that is really necessary. So personally, I'm not a big believer in intuition. I think you can get a gut feeling that an area is going to be important or that something is worth doing, as I just did with metagenomics. I think these bacteria that live with us, they are going to be very important. And we already know they are. There are already enough anecdotes out there to show you that they are going to be very important. If that's intuition, well, so be it. But I think it's really observation, looking around, seeing what's going on, and then trying to put things together and say, well, this would be a good area to get involved in.
Looking at the slide up there about humans and bacteria, what do you think is going to happen with the war between humans and bacteria?
Well, ultimately the bacteria always will win. So I actually have a talk that I give about why you should love bacteria that kind of deals with, first of all, how diverse they are, how interesting they are, how they've gotten a really bad rap because for 100 years the microbiologists have only focused on the pathogens, whereas most bacteria are not pathogenic, they're fine. But I have a slide I show at the end of that talk, which is a picture of Mars. And I say that I hope that before I die, we will discover that there is life on Mars, that it's bacterial, living maybe several miles between the surface, but then when we start sequencing it, we discover it's actually older than life on Earth, and that we came here by Francis Crick's panspermia, in which case it means we're really all Martians, and that would be wonderful. I would like that very much.
Okay, so with that, we're going to end the general program. If you guys are free, please attend the technical lecture at 4 to 5. And thank you all again for coming. If we can give another round of applause for Sir Richard Roberts.
Thank you.